Hello, everyone. I'm Sylvia Lee, and I want to welcome you to the Diatom Web Academy, brought to you by the Diatom Taxonomic Certification Committee of the Society for Freshwater Science and diatoms.org. You can check out the news page on diatoms.org for the schedule of webinar speakers and follow Diatoms of North America on YouTube, Twitter, and Facebook. For those in the Northern Hemisphere, we just had our summer solstice. And with that, the Web Academy will start having webinars on a monthly schedule rather than a bi-monthly schedule. So it will be a bit longer before we see you again, but field work and other summer activities may keep you very busy until next month. On July 20th, the webinar will be about the taxonomy and morphology of a group of canal raphid genera, including Epithemia, Cerarella, Campylodiscus, and a few others, collectively called the keeled diatoms. And please also mark your calendars for the 2021 International Diatom Symposium. It will be an online conference from August 23rd through the 25th, and with special opportunities for early career diatom researchers. Registration will be free for students and early career researchers who are members of the International Society of Diatom Research. So it's a great time to become a member, register for the meeting, and submit an abstract. For the most up-to-date information and key dates, you can go to isdr.org. And now over to you, David, to introduce today's speaker.
Thank you very much, Sylvia. Today I will be introducing Xavier Benito. And I would just like to let you guys first know how I came to know Xavier. It wasn't personal. I more saw him and his efforts to help spearhead the Diatom of the Month blog series, which is a blog series that has been running since 2015. And it's hosted through the International Society for Diatom Research. And I just really appreciated Xavier's enthusiasm for diatoms and really promoting engagement and communication on a slightly less academic level that made it more comfortable and digestible for everybody. And with that said, if you would like to contribute to the Diatoms of the Month blog series, please get in touch with Xavi and head on over to xavierbenito.com if you want to learn more about him. Xavi is interested in limnological, ecological, and biogeography, which we all know diatoms are great tools to use in those fields of research. Xavi is co-chair of the PASS Socio-Environmental Systems and program, and he is also a postdoc fellow at the Continental and Marine Waters program as part of the Institute of Agri-Food and Research and Technology. If you want to find more out about his research, you can go check out xavierbenito.com. And with that, I will let Xavier take it away and talk to about is everything everywhere? Diatom community ecology meets biogeography. All right, Xavier.
Thanks so much, David, for such a nice introduction. I hope everyone can see the screen just right now.
Yes, looks great.
Perfect. Okay. So here you go. Thanks again. I would like to give a big thanks, especially to the Diatom Web Academy team for inviting me. I'm really honored to be here today among many familiar people I saw in the chat. Also, I think this is a really cool series of webinars that help keep the community together in the midst of this trying time. So I really believe this will help to overcome the lack of networking opportunities and somehow democratize a little bit more science outreach and dissemination about diatoms. So today, I would like to talk about some part of our recent research about understanding macroecological patterns of diatoms. So spoiler alert, rather than being a comprehensive presentation on biogeography of microorganisms, especially diatoms, endemism, cosmopolitanism concepts. I will try to summarize using a conceptual framework based on our own investigations that examines community assembly processes and community responses, both from the species composition and diversity side of things to environmental and spatial factors by applying different macroecological concepts or approaches, such as theory of island of biogeography and metacommunity theory. So different levels of higher biological or organization concepts applied to diatoms with the final objective to try to understand whether or not the microbial wall resembles to what we know from the macroecology of macroorganisms. So this is the outline I would like to follow today. So I will introduce the motivation of our work and the study region, so the South American continent and its diatom flora. Then I will go through a kind of macroecology 101 session, introducing some very basic concepts about metacommunity and biogeography applied to diatoms. And then the points 3 and 4 are going to be the meat of the talk, introducing the South American diatom database, which is the main tool we have been using to understand and posing questions about large scale and long-term ecological questions of diatoms. Okay, so I'm pretty sure everyone know these two guys here in this slide. So they are Charles Darwin and Christian Ehrenberg. Back in the days, Darwin recognized somehow the importance of biogeography and history, and how these two factors work together to shape life on Earth. While in South America, Darwin learned about Christian Ehrenberg's microscopic observation of diatoms from diatomids in Terra del Fuego. So they are massive geological formations of millions and millions of diatom frustals. Darwin thought somehow that these geological units or diatoms embedded into it should be old and have wide geographical distributions, because we're talking about Tierra del Fuego, which is down into the southern tip of the South American continent. So perhaps without saying it, the first observations of South American diatoms were somehow related to historical biogeography, which is nowadays a hot topic in microbial biogeography discussions. This is because due to the massive dispersal capacity of diatoms, microorganisms in general, and large populations in cosmopolitan, so existing worldwide distribution patterns should prevail over more restricted or endemic distribution. So in other words, everything is everywhere, the famous Busbecking hypothesis. I don't want to convince anyone here in the room that this hypothesis is true anymore. I convinced myself enough that this is a quite old hypothesis. Since the last 10, 15 years, mounting evidence have somehow falsified that everything is everywhere. Instead, we should talk about everywhere. Everything is different. But I do recognize as well that this question or this hypothesis has been fundamental in catalyzing research in trying to understand how community assembly rules function in the microbial wall, and especially in diatoms. So trying to apply community dissimilarity methods or macroecology, or how community ecologists work with spatial and environmental factors, this plot is a nice example. So things that are relatively closer will be always more similar than things that are geographically far away. So this is something well known in macroorganisms. So they do have biogeographies. In microorganisms, though, no matter the geographical range of a study, some populations may be similar. So the most important implication of this is that environmental factors will always be more important than spatial factors. For instance, dispersal limitation, environmental, spatial barriers such as elevation, climate, etc. When we think about one of the most vulnerable regions on Earth when climate change impact both biodiversity and human security access to water, the tropical South America is one of the most important regions, right? So in the Andes, we found the Andean Paramo, which looks like this picture over here. It always covered by clouds. Their soils are fully covered by water. So these ecosystems, high elevation, wet grasslands of wet grasslands provide ecosystem services to more than 100 million people living not only in the Andes but also in the adjacent Amazon lowlands. This region, though, has been pinpointed by the researchers that it's going to be warming as much as twice as their mid-latitude counterparts. So we might have a problem here when we think about fresh waters and its biodiversity. Lakes and streams of tropical South America are indeed effective systems for examining biogeography and biodiversity patterns of diatoms. The wide latitudinal, longitudinal, and elevation ranges, coupled with diverse physiographic regions and geological substrates, produce notable climatic and limnological variability that eventually can be used to examine or try to tease apart these local and regional factors explaining species composition of diatom assemblages. So the early diatom studies in the continent date from the 19th century, as I said before, by Christian Ehrenberg, and then as time passes, researchers, especially coming from the northern hemisphere, began to catalog and document diatom taxa. Husted in Chile, Joaquín Frengeli in Argentina, Manjón and Servan in the Bolivian Altiplano of Peru, and northern part of Argentina, they all did really important contributions in paving the way for future studies in diatoms. The most comprehensive diatom taxonomic book appeared in 2000 by Rumbrick, and then Metzeldin and Lange-Berthelot continued to document the diatom flora in the continent. However, all of these studies were quite patchy, so and with different purposes. All of them aimed at saying what is actually there, but there has been no comprehensive study to evaluate mechanisms contributing to maintaining what is actually local and regional factors playing diatom community structure.
Hi, Xavi.
Yes.
Just wanted to check with you. Sorry for interrupting.
Yeah, it's okay. No.
Oh,
yeah.
Okay. We're on slide 7 now, right?
Slide 7. Okay, yeah.
Yeah, okay. We weren't seeing the other slide, so I think you're good now.
Okay, cool. Perfect. Thanks, Sylvia. Although southern diatom flora in South America lacks taxonomically calibrated regional data sets, we still can be some we still can do some observations of taxa showing relatively levels of endemisms or even restricted distributions. For instance, here in the Amazon lowlands of Ecuador, several pinularia exist in seasonal flooded wetlands connected to the Napo River. We also know that in Rio Negro, in the Amazon basin, have high levels of endemic species of Eunotia and Actinella. In the Bolivian Altiplano and in Chile and also in Peru, several species have been described as being uniquely found in Lake Titicaca and its Desaguadero watershed, small arrayphyte diatoms as well. So the more time passes, the more studies appeared in documenting restricted diatom distributions. But however, there is still some controversy about whether or not South American diatom taxa are unique in terms of diversity. This is a table I came up. I'm trying to summarize here what is actually found. So as I said before, the study by Rumbrick in 2000, they sampled all the Andean Cordillera from Venezuela all the way down to Argentina. They found almost 900 diatom taxa. Nearly half of them were cosmopolitan. Our own studies were more or less along the same lines, as well as other studies. Perhaps we found a little bit higher number of diatom taxa. We weren't certain about the proportion of the diatom taxa that were truly endemic, but a high number of taxa were not actually, or we cannot describe as or associate to any name. Recently, some studies or some people challenged the view of the Andean diatom taxa as being cosmopolitan, basically because, for instance, Eduardo Morales in 2012, he analyzed a single Bolivian stream sample and he found almost a third of the diatom taxa found by rubric only, sampling a small portion of the area with higher levels of diatom endemics in the diatom list. So potential causes of why we found this discrepancy between observations and studies are not unique of South America. So for instance, early researchers were using the bibliography available at that time and they were doing the best they could. But it is true that these identifications were not based on comprehensive analysis of time material. They simply perhaps transmitted a series of concepts influenced somehow by the taxonomic drift. Taxonomic force feeding using European and North American floras may be another explanation why these discrepancies still exist nowadays. And I believe one of the most important causes to explain the discrepancy is because there is a strong bias in where people were to sample and recollect datum material, right? If we think about Andes, most of the lakes or high elevation streams and rivers are not easy to access, and people, they always go to the easy access to sample. And sometimes these regions are highly impacted by human activity. So we might have a better chance to found diatom communities that are not unique in terms of restricted distributions. But it is also true that only when species taxonomy can be readily determined that a sound ecology is possible, and therefore also a proper comparison with works of similar environments in other geographic regions and comparisons with similar environments can be possible. No big news here, so I don't have to convince anybody in this room about the potential role of diatoms in indicating past and present environmental changes. Perhaps South America has been strongly biased in using diatoms as a paleoclimatic proxies, for instance, using the long-term record in Lake Titicaca for reconstructing past climatic records, glacial, interglacial periods. But again, in terms of modern distributions and higher levels of biological organization of diatoms, we still have a lot of work to do. This is when we started to think about covariation of space, environment, and climate in affecting diatom distribution. So this is nicely represented here with the Venn diagram. So we showed the unique sources of variation by environment, climate, and space. And when these sources of variation shared among them, we might be observing some emergent properties. And this is a question that has been driving my research and our work over the last years to some extent, to try to answer the question if why, or is a species not in a particular habitat only because it was not dispersed, or the environmental conditions weren't suitable for it to grow. So this is quite important. And this is when I would like to give you guys a little bit of the community assembly rules from a very basic point of view. So we have two main paradigms that have coexisted today to explaining the assembling of ecological communities. Very simple. This can be more complicated, but to put it simple, we have the island perspective that focuses on processes such as immigration and extinction. And then we have the niche perspective, in which we have processes that emphasizes species autoecology and environmental filtering. So along the environmental gradient, we observe species substitution or species change in their relative abundance, while, for instance, pH or nutrients change. These two paradigms or perspectives are not mutually exclusive, and they might be mediated somehow by the dispersal abilities of diatoms. One might think that the diatoms do not have dispersal abilities, but if we distinguish or we set apart plantic versus benthic species, plantic species will always be more easily dispersed through the water, versus benthic species due to different attachment modes can support more the physical disturbance of the environment. Lakes have been regarded as ideal models for studying the influence of environment and spatial effects on species composition, because they are not really isolated in the landscape, but rather connected somehow by all the hydrological network. In this context, the metacommunity theory, which recognizes a set of local ecological communities that are connected by the dispersal of potential interacting species, provide a strong framework to generate hypotheses about the underlying mechanisms that control the assemblage of communities. This is particularly relevant for the case of diatoms, because if we think about, or we might expect species to be strongly affected by the topological distribution of aquatic habitats and the different aquatic dispersal pathways connecting them. And then once the diatoms appear to be or join the local community, other factors such as niche assembly processes might be affecting them as well. The metacommunity theory falls within the larger biogeography concepts or sets of relationships that at the end, this is exemplified in this conceptual framework that we published with Sherry a couple of years ago in a book chapter about neotropical diversification. So we have the regional species pool, in which all the species must go through a series of nested filters to end up joining the local community. So bear in mind that these nested filters are hierarchically organized through a niche and dispersal-based process. For instance, in lakes, the local community might also be changing depending on the benthic and the pelagic community. So the benthic community will always be affecting what is actually found in the water column, right? And at the end, of course, species might be deposited to fill up the lake sedimentary record. I'm not going to talk about paleolimnology today, but it's also important to have in mind that historical processes are also processes that affect the biogeography of the species through, for instance, priority effects, which were the previous communities that were allowed to colonize new habitats that might have an effect for later immigrants into the community. So central to test all of these ideas is our South American Diatom database, which is shown here. I'm going to talk about it in a minute. But basically, we have lakes, streams, and rivers located all over the Andean Cordillera, right? And some sites located in the adjacent Amazon lowlands. We also have environmental data associated with the sites, limnological records, and most importantly, perhaps, this database is fully available in the GitHub. I don't know if anyone is familiar with it, but it's a kind of open access repository in which people can download data sets. It has some functions to visualize data and clean some data sets, etc. So people are more than welcome to download the database and provide comments. I'm always happy to initiate collaborations from people that might have data sets that may contribute into this database. So this slide aims at summarizing a little bit the conception of this database and how it has been evolving over time. It wouldn't be possible to build this with help and collaboration with many people. I tried to put their headshots here at the bottom, but I'm pretty sure I lost someone, so my apologies. We started this working with 3 different sources of data. So Diatom counts from published works, and also Diatom material that people send us to analyze. We take photos, we photographed all the species or forms and varieties, and we made some Diatom voucher floras for the initial assembling of the database. Then we came up with the most tricky thing, but at the same time more exciting exercise of taxonomic harmonization. We think about different collections, different analysts. I'm going to explain a little bit more what we've done in this site. So after the taxonomic harmonization, the first version of the database was published and is available in a series of Excel spreadsheets in Riot. The second version of the database was a little bit more extended in a space with new sites and new projects coming in. And the most recent updates were the two Shiny applications for database visualization and see exactly where and when Diatom taxa is present in the different locations. And recently we have started to talk about the Neotropics database developers to upload this database into this larger community-driven community of ecological practice that is Neotropics. And also it's going to be relatively more easy for people to contribute with their data in a much more dynamic platform. Okay, so the overview of the database. So we have a total of 326 unique sites distributed in 26 regions. The temporal period of observation ranges from the 80s to present day, let's say 2017, 18. Extent, we have samples from almost sea level to up to 5,000 meters above sea level. Along with diatom counts, we also have environmental data associated with it. The diatom counts data, almost, I would say it has 20% of counts that have presence absence data. And as I said, the environmental data is highly heterogeneous, especially for the water chemistry component. As you may imagine, different collections have different purposes, different objectives. So it's really tricky to have a consistent set of variables characterizing the water chemistry. But still, we were lucky enough to have at least pH, conductivity, water temperature, some nutrients data relatively consistent across the database entries to test about our hypothesis. The physical context and geo-climatic context were mostly obtained using GIS layers of information. So out of the almost 400 unique sites, most of the sites belongs to lake habitats. We also have rivers and streams and some wetlands. Sample types belongs mostly to modern, let's say, phytoplankton, periphyton samples, surface sediment, and also core tops from paleoclimatic studies. The year, so the temporal coverage of the database, as I said, ranges from the 70s, 80s, all the way to present day. It doesn't mean that we have time series of data. I wish, or we wish we have replications of or temporal data to test temporal evolutions, for instance, of the communities. But it's quite heterogeneous also in terms of the temporal coverage. And these are two screenshots about these two Shiny apps that we developed to visualize what is actually the database and the distribution of sites. So for the database exploration, for instance, we have several tabs summarizing each region, which diatom tags are present there, the environmental variables, the distribution of the variables through a series of box plots, ecological groups, benthic, tychoplantic, saline, diatom taxa, etc. And also we have the taxa distribution. So for each diatom taxa, we have where exactly it's found with a semi-categorical classification of relative abundance. So a key component, so let's talk a little bit more about taxonomic harmonization, which was quite an important key point in building this database. Unfortunately, only for a small set of sites, we were available for accounting. So let's say the original analyst performed the analysis, and then Melina and I did the second analysis, and we compared this and tested to what extent taxonomic harmonizations improve or try to reduce the analyst bias, which is quite an issue when we build large biological databases. So these are the main steps that we follow for taxonomic harmonization. And then when we have this, we compare the two data sets with an NMDS, a non-metric multidimensional scaling, which plotted the ellipsoids to show the overlap of analysts before and after the harmonization. And the results show how samples relatively become more similar after we harmonize the data sets. For instance, aggregating varieties and forms to nominal species, updating for taxonomic synonyms, and so on. Okay, so from now until the rest of the time I have left, I would like to present some of the outputs of the database and the studies that we published using this tool, beginning from species level, ending up to more higher levels of biological organization of diatoms. So we began trying to answer the question to what extent plantic diatom species were widespread across the continent or mostly located in tropical Andes. We focus on plantic diatom taxa because they are highly sensitive to responses of thermal lake structures such as warming and nutrient inputs. And our results show that there exists a broad elevation and latitudinal distribution of the main plantic diatom species in our database. Then we went a little bit further and we modeled the species distribution using a logistic generalizative model using presence absence data, because we didn't want to throw away that portion of database entries that do not have relative abundance. And for some species like this Costella steligera, for instance, conductivity was a strong limnological factor while controlling for water temperature and pH. Some Alaucosaura are strongly affected by water temperature as well. Then the next question was to try to answer to what extent a regional group of lakes based on their environmental similarity may act as metacommunity units, right, for lake benthic diatoms. We began summarizing the environmental variability of lakes using a principal component analysis, which is shown here for PCA axis scores of the sites and then the variables. We saw how lakes are arranged pretty nicely along a latitudinal gradient, right? In this latitudinal gradient, limnological factors are embedded into it. And the second axis of major variability was summarizing variables associated with what we called landscape configuration, including variables such as aquatic connectivity, lake area, terrain roughness index, which is a kind of variable that tells you how complex the landscape it is, and elevation. Connectivity and aquatic habitat and percentage of aquatic habitat were quite interesting, are important. I'll explain you in a minute for predicting diatom species richness. Then we grouped using PCA axis scores to come up with 6 total different lake clusters or lake districts, which again serve to compare with the diatom data using an NMDS. And here, exactly the same main axis of variations arose. Here, this is not a constraint ordination analysis, which means the samples or the sites are freely distributed based on the environment, the diatom similarity among them. And we added environmental variables, and the same latitudinal and landscape configuration emerged. So we wanted to test to what extent the environmental matrix and the diatom matrix match together, and if they are correlated, and they were. So we proceed with the basis that diatom biogeographic patterns somehow emerge due to 3 main reasons. The first one, as we observed, regionally distinct floras with significant association with geographic group of lakes were observed. We also found latitudinal gradients of species richness and community structure, which is quite quite important. If we think about latitudinal gradient of richness is one of the most conservative and well explained and documented pattern on Earth, in which plants, mammals, birds all follow this more or less the same pattern over the latitude. And also using a variance partitioning analysis, I don't have time to explain the details of this statistical approach, but it tells you the unique and shared proportion of variability explained for your diatom or your biological data set. So broad scale spatial factors, which means geographical distance, and geo-climatic factors, climate, elevation, and so on, explained more unique and shared percentage of variability than limnological factors. So there is a joint contribution of environmental filtering and dispersal dynamics affecting diatom communities composition in tropical South America. A natural follow-up question from here was, okay, so to what extent local and regional factors, let's say niche and island paradigms, structure diatom metacommunities within lake districts instead of across lake districts? And if there is any metacommunity process behind the latitudinal gradient of organizations that we observe in community structure. So we performed the same type of analysis, variation partitioning for each lake cluster individually. And unfortunately, there is no clear pattern. For instance, let's say, so these are the lake clusters ordered by latitude, and the larger the bar is, the more percentage of variability is explained by the different types of predictors. So we have geo-climatic predictors, geographic distances, limnological predictors, and the shared proportion of among the sets of predictors. And the lake clusters are arranged following a latitudinal gradient. So we did not observe a clear pattern. For instance, we might thought in the equatorial region, environmental characteristics tend to be more constant all over the year, and we might think that environmental factors are not as important as regions located down the Andean Cordillera, for instance, in the Bolivian Altiplano, which seasonality of precipitation and other type of limnological characteristics are more variable in nature. We try to put all this unique and shared proportion of variability into environmental heterogeneity and spatial extent context. So for instance, lake cluster number 3, which is the Brazilian and Bolivian lowlands, it is the only one in which the limnological component explain more variability than the spatial factors. And this might be explained because the higher environmental heterogeneity a lake cluster has, more ecological niche diatom communities may occupy, diatom species may occupy, right? On the other side of the history, for lake cluster number 4, which is the Peruvian Andes, it is the one that has the largest spatial extent. We might expect more control of geographical distances because dispersal limitation in geographical barriers and so on. So this is still something that we want to test more hypotheses why there is no clear meta-community process behind the latitudinal gradient of organization. Some explanations might be, as I said before, different dispersal abilities of diatoms might be also playing a role. So we did publish that, but I did not include it in my talk today. But if we do the same variation partitioning approach, deconstructing by ecological guilds, for instance, we might observe other emerging patterns in which the relative role of spatial and environmental controls explained more or less variability in the samples. So a prior examination of biogeographical patterns of diatom communities in the first version of the database showed that geographic regions within Ecuador had relatively more shared diatom species than within other regions of the Central Andes and adjacent Amazon lowlands of Peru and Bolivia, despite similar geographic distances, right, between highlands and lowlands. So we thought, okay, there might be other factors playing a role here to explain why the equatorial diatom forests are relatively more similar than the ones located down the latitudinal gradient. So we built a generalized linear model trying to understand which are the main environmental predictors and geo-climatic ones, explaining diatom species richness. If we think about the importance of aquatic connectivity, for instance, in shaping a species composition and richness, this is a fundamental component of the theory of island biogeography. That is, species number at equilibrium, so colonization versus extinction rates, is a function of habitat isolation and size. So here what we found is that nicely the connectivity, which is the number of water bodies within surrounding the lake, let's say, or the site of a study, have a positive effect in explaining diatom species richness, which suggests somehow that diatom species richness increase with water connectivity, and on the other side, elevation has a negative effect, explaining the diatom species richness, which again suggests that a decrease in the remoteness of the system increases species number, which more or less or somehow match with the expectations of the theory of island of biogeography applied to benthic diatoms. We wanted to also explain or see to what extent diatom chains using the diatom database, or so here a little bit of paleo limnological studies, not too much, but to give everyone a little bit of taste about temporal change. To what extent diatom change is widespread in tropical Andes during the Anthropocene. So in this plot, we connected 2 time points, 150 years ago and modern times. And the lakes, the 2 time points are connecting the lakes. So the longer the lines, the more dissimilar the diatom communities are. So we plotted and we compared that lakes located in the Andean Plateau of Ecuador and lakes located in the higher Andes. And in general, except of 2 sites, there is no much change going on, right? So we thought why, or we posed the question why some systems are more prone to be stable than others. We started to posing questions about ecological resilience and recovery of communities after following stressors. Some people in the Andes have concluded, for instance, that diatom communities may have crossed climate-driven thresholds based on observation of spikes in increase in Discostella strigida in lakes located in Ecuador. Okay, but if we are able to extend the temporal window observations over the last 7,000 years, for instance, analyzing the same diatom species, but in other lakes with different nature, the same species may follow periods of near extirpation from the record and then come up again to be a dominant component of the community. Exactly the same pattern might be observed with a longer temporal time, with different lakes, and so on. So we were highly attracted by terms such as community assembly and disassembly over time and applying ecological resilience questions. And what we mean by ecological resilience. So this is nicely exemplified here by the ball and cap theory. So an abrupt change. So ecological resilience theory, for instance, on the left, a high resilience state, right, is depicted by a deep cap so its basin of attraction is relatively deep, right, and the ecosystem, which is characterized by the ball, returns rapidly to an equilibrium after a perturbation. In contrast, if we are in a low resilient state, every time the ecosystem suffer a perturbation, it may be more prone to cross a critical threshold and enter into an alternative stable state. So the basin of attraction is much more shallower. All of this theory has been developed using monitoring data and manipulating lakes, for instance, adding nutrients until the shift was observed. I believe, or I'm a strong believer that the same reasoning can be applied using proxy data, and not only one, but multiple basins of attractions can be observed over time to get a wider catalog of disturbance and ecological responses relationships to better understand ecology. So this is exactly what we did. I think I have enough time for go to the last slide. We modeled Diatom community time series using nonlinear time series methods such as GAMS. And we calculated rate of change. So rate of change is usually depicted as one of the measures to capture this resilience change or abrupt changes in the community. We modeled this using a series of 4 lakes in Ecuador from more to less remoteness along a gradient of remoteness. And we observed that more or less, or in general, except for Lake Yavicu, which is one located relatively close to human settlements, and Lake Yaguarcocha, the 2 lakes located up to the mountains in the high Andean Paramo, there appears to be any period in time in which diatom assemblages suffer an abrupt change. Even we know that they suffered some well-known climatic and human perturbations in the lake catchment records. These are really nice time series analysis to answer questions about change in ecological resilience using diatoms, but it doesn't tell you about what is the nature of change. So we can compile or use other type of statistical techniques to answer this question. So for the same 4 lakes, Piñán, Yaguarcocha, Fondococha, and Yaviucu, so we have the modern distribution of diatoms from the tropical South American diatom database, which is depicted by the gray tiny dots here. Then we have the fossil core trajectory for each lake. And on the top of that, we add another layer of information, which is isolines depicting changes, in this case, a total phosphorus. So every time that the fossil trajectory may cross one of these thresholds, it might be informative that the community is affected by this environmental variable. We can add as much as variables as we want, or testing different hypotheses. For the case of Yaviucu, which is the one that we observed an abrupt change over the last 150 years, indeed, it crossed several times, several times thresholds of total phosphorus. So this is some questions that we might also pose using the tropical South American diatom database, not only from a spatial perspective, but also a temporal one, and answering questions about, as well, biological naturalization and so on. Okay, so I guess I'm almost heading to the maximum time of my talk, which is 45 minutes, I'm afraid. So these are some of the open questions that I came up while I was preparing this talk, and I will be more than happy to talk with you about some of them. For instance, I know I did not go into much detail about the proportion of cosmopolitan versus restricted taxa using our own data. I think we are not in a strong position to draw clear conclusions about this topic, but as more studies appear in terms of, for instance, using molecular tools, we were being more certain about what is actually unique from the continent, and then we can start making comparisons with flora outside the Neotropics, such the all Arctic. Another really interesting topic of research that I'm keen to pursue is what is the role of functional biogeography in neotropical diatoms. For instance, comparing, making hypotheses about the different ecological guilds may have different dispersal abilities, and hence some other biogeographic patterns may emerge as well. Okay, and with that, I will be happy to take any questions, and I'm strongly thankful to all my group of friends, collaborators. Some of them are here today in this talk, so I'll be really happy to answer questions or begin a dialogue about how we can better use diatoms to answer macroecological questions. The slides and references are fully available in this link, so people can do whatever we want with them. And yeah, thank you.
Thank you, Xavi, for a really great talk. And if people would like to put in their questions into the chat or raise your hand, we can have Xavi answer your questions. While people are getting ready, I wanted to ask about dissolved organic carbon. Was that one of the factors that you examined?
That's a really nice question, Sarah. Thanks. So we, unfortunately, dissolved organic carbon were available for a very tiny set of sites in Ecuador, especially. So Ecuador is the best represented region in terms of coverage of environmental variables for studying their effects into diatoms. For instance, I don't think I have enough information to answer the question, but it's going to be for sure a strong effect, for instance, in lakes located down in the Cajas National Park in Ecuador. Lakes located down the lake chain are receiving more dissolved organic carbon from the Paramo soils, which are
yeah, I see.
Getting into,
is it Melina says we have it for Cajas? Is that or DOC?
Yeah,
because I imagine it would be such a strong influence in warm regions.
Absolutely. Yeah.
Okay,
again,
and Mark comments that you show clearly how recent diatomists are recognizing exciting patterns of biogeography in South America. Did the early diatomists like Franguelli and Ehrenberg acknowledge these patterns there in South America?
I don't know about Ehrenberg's observations or conclusions drawn from his early studies, but for instance, Joaquin Franguelli, he did acknowledge biogeographical, let's say, or restricted at least distribution of diatoms in Tierra del Fuego, which is outside the tropical region of South America. But unfortunately, Franguelli's work is quite old and is all based on, his drawings, and many people has contested or challenged even the true identifications made by Franguelli. But I think, well, I don't know exactly what's going to be the best way to answer this potential or early biogeographic patterns recognized by diatomists, but I guess a nice way would be to rework or work with the original work by Franguelli, which was a pioneer in studying South American diatoms and see what is actually was true endemic or not in his observations. Yeah.
David says, great talk, Xavier. Thanks for sharing. Was lake depth or lake mixing quantified or considered in your species distributions?
Thanks, Dave. So lake mixing, unfortunately, not for the most part of the results I showed you guys here today. We are just working with a new study from, again, the Cajas National Park of Melinas is leading very nicely, but lake depth, for instance, was included into the analysis and was part of this landscape configuration or physical factors explaining diatom distributions. Of course, was quite an important factor, but lake mixing, unfortunately, we don't have enough data for including this variable into the larger set of analysis. But this is a really nice question. Yeah.
Good questions. Karthik asks, how do you deal with the unknown and new species while doing your ecological analysis? As those need to be assessed to the best possible level for meaningful interpretation.
Yeah, thanks, Karthik. Yeah, well, so yeah, this was actually the most important issue or challenge that we have with unknown species. So think about that. I tried to make it clear at the beginning of my talk that this is not a really a truly or comprehensive biogeography study that we did. Instead, we tried to apply datum community analysis to understand spatial distribution. So at the end, it doesn't make sense to put a known or not, because we just want to have a name at the end for the diatom taxa that we are analyzing, and we have to make sure, of course, that this is different from other taxa. But of course, as long as we might have in the future, maybe and hopefully new observations, and we might be certain about this high percent of unknown species that we have right now in our database, I'm pretty sure that these patterns are going to be different, even with more, let's say, restricted distributions, or this geographical distribution of the species would be even more stronger.
Yeah, and Karthik has a follow-up to that, which is how helpful is the voucher flora?
Yeah, it's quite helpful. Yeah, it's quite helpful because once you have the voucher flora, you can share it with other, let's say, taxonomists. I'm not a taxonomist, so people can help if whether or not these taxa are more or less or familiar to them, and they can help us really identifying or trying to disentangle the morphological variability of some species. Yeah, so it's really helpful.
Leila asks, thank you. And have you distinguished closed basin lakes with only, well, I'm not sure if that's closed basin, but with only rain input, and those with spring sources? If so, how do you distinguish the climatic signal in those types of lakes?
I'm not quite sure if I understand correctly the question. So I guess, yeah. So okay. So let's imagine I'm understanding correctly the question, and I'm going to answer the best way that I can. So closed basin lakes are ideal systems to test these hypotheses of island of biogeography, because you don't have inputs and outputs on. So everything that's happening into the lake is the product of, as I said before, the benthic community affecting the pelagical the pelagic community, and then all, for instance, lake level changes, it's going to be a product of precipitation and evaporation changes. And this is quite nicely recorded in the sediment record to reconstruct climate changes. I don't know if that answered your question, how we can distinguish the climate signal in this. So the climate signal in closed-based lakes are quite clear. It's even more tricky with lakes that have input and output, but at the same time are even more interesting from the metacommunity point of view, because they are connected at the landscape level, and everything that's happening up to the lake chain is affecting somehow by immigrants and diatom frustals into the lake. Sorry if I did not.
Yeah, Leila, if you have a follow-up, go ahead and put that in, or just chime in. And Mark asks that first comments that many of us are involved in trying to develop scientific community resources, such as Neotoma and Diatoms.org. What are the challenges that you're facing in developing a South American database? Is it funding, participation, accessing data? What are your challenges?
Yeah, that's a really good question, Mark. Thank you. Perhaps lack of time or quality time to spend in really. So the most important challenge I found, or we found in developing this database, is the high variability of datum collections that we put together, because each collection has different analysts, different purposes of a study, and so on. And the more we advance into the chain of process, we might be finding more idiosyncrasies about that collections. It might be tell us Now that was from the phytoplankton, but then we found out was periphyton, and then we need to take into account all the sources of variability to really to accommodate the metadata. So the metadata is also a really important part in building a database, even more than the data itself. Funding is also a really challenging point of view. If we don't have funds to continue research, it's going to be an end product or an end project, and it's a pity after spending so much time and effort that this is going to be end. And this is why we are trying to make it accessible or upload the database into Neotoma, because it's a really big project. There are people investing time and money into making a really useful and dynamic database that is at the end available and open access for the community.
Well, thank you for your work on that and contributions to our broader community. I know that that's really substantial effort, and we appreciate that.
Thank you.
So we are at the top of the hour. So I'd like to thank you, Xavi, for presenting in the Diatom Academy. And we're going to have a bit of a next break. A month before our next talk, Tuesday, July 20th, Sylvia Lee will be talking on the keeled diatoms. So I hope everyone is well and safe and continues to connect and do good work on diatoms. We'll see you next time. Bye.
Thanks so much.